webinar from the Microfluidics Innovation Hub. Thank you very much for joining. And today our presenter is Dr. George Tsekenis from BRFAA in Greece, which is the Biomedical Research Foundation Academy of Athens. And the presentation today is titled Functional Nucleic Acids, Unleashing Their Untapped Potential in a Diverse Set of Applications. And George is an expert in aptamers, functional nucleic acids, biosensing, and surface functionalization. He will be talking about aptamers, introducing us to what they are. Will be a comparison to antibodies in specific applications, the potential that aptamers have, their benefits, a little bit about assay development and optimization, and also applications in biosensing and specifically towards microfluidics, which is the focus of the Microfluidics Innovation Hub. So George, may I ask you to
Share
put
my
forward
screen.
the slides? Yeah, yes, please.
Cool.
So just now that we have everyone here, just to say that this webinar will be recorded, and April will provide with a link to access it later. We will have roughly 40 minutes of presentation, 40 to 50 minutes of presentation, followed by a Q&A session. I just realized I have not introduced myself. I'm Devesh. I'm the Technical Program Manager for the Microfluidics Innovation Hub, and I will be moderating the webinar today. Okay, so let's get started. I'm just going to give you a very brief introduction to the Microfluidics Innovation Hub and next-gen microfluidics, just to orient you regarding who we are and how we work. So George, can I ask you to flip to the next slide? Okay, so in order to understand what the Microfluidics Innovation Hub is, you would need to know what is next-gen microfluidics, which is a Horizon 2020 Open Innovation Testbed consortium comprising 21 companies across Europe. The companies provide expertise and services in the development and manufacture of microfluidic devices for diverse applications, especially, let's say, in vitro diagnostics, lab on chip. I'm sure you're familiar with those terms. And in this case, the Microfluidics Innovation Hub acts as a single entry point towards the combined technologies, services, and expertise of these 21 companies. And together, we comprise currently the worldwide biggest platform for upscaling, developing, and testing microfluidic devices. George, may flip to the next slide. So just to orient regarding what's the topic of today's presentation, we consolidate the services, expertise, and technologies offered by our partners in the consortium into a value chain in the development of microfluidic systems. Often that starts with the development of an assay, which then gets transferred onto, or let's say miniaturized onto a lab on chip or microfluidic system, for which microfluidic chip, for which usually this can also then be interfaced with a readout device, may it be for optical detection, electrochemical, etc. So we, together with the 21 companies, we comprise services along this whole value chain. George, may you move to the next slide, please? Over here, you can see roughly how the companies that are part of the consortium fit into the value chain. And today we will be focusing on BRFAA, which you can see right at the bottom left corner, is particularly specialized and has in-depth expertise in the development of assays. And we lend, George lends his expertise, technologies, and services to the consortium in the form of developing our demo cases, but also towards projects that we have with customers that are developing microfluidic devices. So without further ado, I will, after the presentation, I will give some information regarding how to access the services that we offer and also the funding that we offer alongside the services. But for now, I will give the floor to George to talk about aptamers and functional nucleic acids.
Thank you,
Thank you
Dinesh.
very much.
Thank you. So I hope you can hear me well, all of you. Thank you very much for joining, and my apologies for those of you that joined 2 weeks ago. In the first time that we had the webinar scheduled for, unfortunately, due to some unforeseen circumstances, I couldn't join. So hope this time round it works fine. So as Dinesh said, I'm George Tsekenis and I'm heading the Applied Biophysics and Surface Science group at the Biomedical Research Foundation of the Academy of Athens. We like to call it Academy for short because our name is just too long. And I've got a couple of slides at the end of the presentation to introduce you to to our expertise basically and what we're doing here in our laboratory. So the presentation today is focused on something that is very close to my heart, functional nucleic acids, aptamers and DNA enzymes. And here is a brief overview of the contents of the presentation. First of all, what are these functional nucleic acids? What are the different types of, and how do we actually get to select these sequences? Then the usual comparison. I'm sure a lot of you have seen these nucleic acids, these functional nucleic acids being compared with antibodies. So the big question is, are they better to antibodies or on a par or not? So I'm gonna try to give some answers on this topic, or this dilemma, on what one should choose when it comes to designing an assay or for a specific application. Then I'm gonna discuss some challenges that aptamer research and aptamers as molecules have when it comes to their applications, and ways forward. How can we circumvent, how can we overcome the challenges faced by aptamers? So then very, very briefly, I'm going to talk about some applications of functional nucleic acids. And lastly, I'm going to present a couple of slides on our laboratory and our activities here in Athens, Greece. So starting off with the different types of functional nucleic acids. Now, you all know that the discovery of DNA, or actually the discovery of the structure of DNA, is relatively recent. It only happened, in 1950s. So talking about functional nucleic acids, or knowing that nucleic acids can have more as a function other than carrying the genetic information or participating in the cellular processes of transcription and translation, is even a more recent development. Now, such functional nucleic acids exist in nature. For example, the ribozymes were discovered in 1982, and only 1982, and that was the first time that we realized that a nucleic acid in specific RNA can have catalytic activities. Up until then, we were only aware that enzymes, therefore proteins, can have such a role. Now, based on this, the whole hypothesis of how life actually began got formulated, considering that RNAs participate and play a vital role in regulating specific cellular activities and being able to have a catalytic role. So that was a revolution in itself, and being aware that RNA sequences can have a catalytic role. And then what was even more of a revolution was the discovery of riboswitches, which only happened in 2002. Now, riboswitches actually recognize small molecules, and they have some sort of regulatory action on gene expression. So that was the first time that people actually realized, scientists realized, that recognition of a specific sequence can actually change the structure of a nucleic acid and trigger a downstream response. However, even prior to this discovery of the riboswitches, people have already started thinking of aptamers and aptazymes, and all these nucleic acids which are synthetic and can recognize specific sequences. So it was in 1990 that the first aptamer was actually synthesized and selected from a library of nucleotides, as I'm going to show you in a bit. So it's all very recent. It's a very exciting field because there is a lot of things that have not been discovered, a lot of things that we don't know just yet. But first of all, what are the different categories of functional nucleic acids? As I already introduced, there's those that are catalytic, that they exert some sort of catalytic action onto themselves or to sequences that they hybridize with. And this, depending on whether they are ribonucleic acids or deoxyribonucleic acids, they are termed DNA enzymes or RNA enzymes. The most usual, the most well-known ones being the ones that recognize specific heavy metal ions, and in doing so, they exert a catalytic action. And then there's the large category of non-catalytic ones, which are the well-known aptamers. They can be both DNA and RNA. There is a lot of debate as to which one is better. We know that RNA naturally has the tendency to acquire more complex structures, but by all means, I mean, they cannot be used exactly interchangeably, but depending on the application that you have, you can use either. And then there is a combination of this, the RNA enzyme plus the aptamer. So the aptamer gets to recognize a specific target molecule, and then that triggers a change in the rest of the sequence, which exerts then a catalytic action. So the list and the combinations are endless. It is the very nature of nucleic acids that allows us to manipulate them in such a way to extend the sequence, to combine different tasks or different activities in a single molecule, which makes them so fascinating and so versatile as molecules when it comes to developing an assay, or for many other different, in biosensing, or for many other different applications, as I'm going to talk in a bit. So how are they selecting? Their selection is artificial, which means that these sequences, although we got inspiration from nature, do not exist in nature. So it all starts with preparing a library of random oligonucleotide sequences. You can see it here. Sorry, I don't know if you can see my cursor. Where you have a random region of nucleotides. This can be 30 or 40 nucleotides in the middle, along with two primer regions, two constant regions at either end of this random region, which are important for amplifying the sequences that get bound to the analyte. So the idea is, in traditional SELEX stands for sequential after the amplification selection of sequences. The idea is that you immobilize whatever the target that you want to recognize is on a solid support. This can be agarose beads, this can be Cephorose beads, or it can be nitrocellulose paper. It really depends on the type of target that you have. And you incubate this immobilized target molecule to this random DNA library. Now, these oligonucleotide sequences acquire some 2D and 3D conformations, which allow them to bind to the analyte of interest, which is immobilized on this solid support, selectively and specifically. Now, you can remove ideally you would like to expose the same library to the target, for example, that is very similar to the one that you actually want to have in your application, so that you counter select the ones that bind to that target as well, so that these sequences are removed, or you can do this negative selection after the positive selection. In all cases, the idea is that the sequences that get bound onto the immobilized target get isolated, whereas the rest are thrown away, are removed, so that you can be left in the end with a number of sequences that bind specifically to your target analyte. Then you elute these sequences from the bound target, and what you're left with are the sequences that all of them, if you remember, have constant primer regions, which can be used to amplify the sequences and use them in a second and a third and a fourth. Usually it takes up to 10 or 15 rounds of selection to actually end up with a number of sequences. It's never just one sequence that recognizes specifically and selectively and with good binding affinity your target of interest. What happens is that in each selection round, you make the conditions more and more stringent so that you get rid basically of sequences that do not bind that well or with that high specificity or without good binding affinity your target analyte. So this is the standard SELEX workflow. And as you can understand, this is quite laborious because it usually takes up to 3 months or 4 months to end up with a number of sequences. It's never just one sequence that bind to your analyte of interest. Then what happens is that you have to validate the sequences or compare them, align them, so that you come up with some common shared structural motifs, so that you identify the part of the sequence that is important for binding to the target analyte. Now, over the years, and as I said, this is a very recent development, so things change on a yearly basis and improve. There's been lots of variants being introduced to simplify things, to optimize things, to make the selection better and less laborious. And it really depends on the type of target analyte you have. So I'm not going to discuss extensively the different types of SELEX when it comes to where exactly do you immobilize, for example, the target analyte. So a very recent development is the magnetic bead-based SELEX, where the target analyte, instead of just being immobilized on some agarose beads, they get immobilized on magnetic beads, and therefore the actual pull down assays and so on are simplified and sped up. Of particular interest is the introduction of, for example, of capillary electrophoresis, which you can see here. Now, the interesting thing about this variant of SELEX is that neither the target nor the actual library is bound on a solid support. Now, this is very important because we all know that once you bind an analyte or a target library or the library onto a solid support, then there are some structural changes introduced. Or when it comes to small molecule analytes, then there aren't that many sites that you can use on the molecule itself to immobilize it. So you have to modify it, which means that you're changing the native conformation, you're changing the molecule itself to have it on a solid support. So what capillary electrophoresis has allowed is the selection of sequences without the need for immobilization, so at their native state. So this is a very important advancement when it comes to selection of sequences. Another thing is that sometimes you cannot immobilize the analyte simply because it is just too small, or it doesn't offer the right functional groups that you can use to immobilize it on a solid support. In that case, a technique called capture SELEX has been developed, where the actual library, this random library of oligonucleotides, is immobilized with the use of a complementary sequence on a solid support. So the things have been reversed. It is the analyte that is in solution rather than bound on the solid support, and the library now is found bound on the solid support. That makes it easier to select sequences that bind specifically to analytes in solution, which are just too small. For example, think of heavy metal ions and things like that that simply cannot get immobilized. Obviously, there's a lot of other types of SELEX, for example, whole cell SELEX, for the selection of sequences that bind specifically to epitopes or antigens on the surface of cells. And also next generation sequencing CELEX, which is an advancement that can get combined with the rest of the variants of CELEX. This simply has to do with sequencing all of the sequences that become available in each round of CELEX. Traditionally, what we do is we only keep and sequence to find out, the actual order of nucleotides in the selected sequences at the end only of CELEX. So in round 10 or round 15. Now, that means that we discard lots and lots of sequences that could be useful. I don't want to, go into a lot of detail as to what each round entails and what the sequences could be. But the idea is that a lot of times, simply because you have this PCR amplification step, which also complicates things, and you also need the primers to perform this PCR, there is a lot of potential when it comes to CELEX that is being lost simply because of the requirements of traditional CELEX. So this next generation sequencing allows us to sequence not just the sequences that arise at the end of the selection rounds, but in each round. And most importantly, the fact that you can sequence oligonucleotides at very low concentrations makes it possible in some occasions to get rid of the PCR step, which means that you don't need the primers, which means that you are able to recover sequences that would otherwise get lost. So this coupled with the capillary electrophoresis is a very important advancement when it comes to being able to select sequences for molecules at their native conformation and maintaining all the information, all the possible sequences, some very interesting sequences there. So moving on to the advantages. I put advantages over antibodies, being full aware that I don't like this title. But everybody's talking of aptamers as being the counterparts or the rivals, if you want, of antibodies. So I'm sure you've seen lots of tables. I mean, if you Google aptamers versus antibodies, you'll see lots and lots of tables where they compare them as molecules and they compare their properties, which make them more or less useful for the downstream applications. So, being biorecognition molecules or being able to bind specifically and selectively to a target analyte, what you want from both of them is to have a high affinity for the ligand. And that is something that they both have. Both antibodies and aptamers have high affinity for their target analytes. When it comes to selectivity for the ligands, antibodies, having been naturally selected over thousands and thousands of years, there is no doubt that their selectivity for the ligand is high. Now, aptamers, I've put here a question mark. It is high when the analyte that they're meant to bind is presented to them in a matrix that is not too complicated. So, for example, if you have an aptamer that is meant to bind a toxin, like aflatoxin or ochratoxin, for example, for a food application, then there is no problem. The affinity is high, the selectivity is high. But if the same toxin is found in a much more complex matrix, for example, if for some reason you wanted to identify this in blood plasma, let's say, then it would be a whole different story. So for most purposes and the way that the samples are pre-treated or are presented to aptamers allow for this high selectivity, but still they don't really compare to antibodies in that they are not generally selective. They're only selective under specific circumstances, which in most occasions are good enough for what we want the aptamers for. Another very important advantage of aptamers is that they can target the range of target analytes that they can bind to is a lot wider in comparison to that of antibodies. Now, antibodies can only bind to specific epitopes on antigens, and they cannot. We cannot have antibodies against things that don't trigger an immune response. So, for example, there isn't an antibody against glucose. There is an antibody against the enzyme that catalyzes glucose. So if you want to develop a sensor, for example, for glucose, what you usually search is glucose dehydrogenase and not glucose directly. However, aptamers, they don't have to be developed and they don't have to be selected against something that causes an immunological response. That means that we can have aptamers against heavy metals, as I said, small molecules like glucose, lactose, and things like that. And that makes them a lot more versatile when it comes to the types of targets that they can go for. Now, cost-wise, obviously the development of antibodies is a lot more expensive than that of antibodies, than that of aptamers, because it involves the use of animals. Another thing is that depending on the batch, there is a lot of variation between the antibodies coming from different immunization rounds or batches. Aptamers, on the other hand, since they are not just synthetically or artificially selected, but also synthetically synthesized, they're always consistent, and they are considerably cheaper than the antibodies. A further advantage of antibodies is that they're a lot more stable than antibodies, of aptamers, sorry, is that they're a lot more stable than antibodies. As you're well aware, aptamers are nucleic acids, and therefore they can withstand extreme conditions, be it the presence of organic solvents, be it pH values, solutions with pHs that are well beyond that of physiological range, and also extremities when it comes to temperature. We actually take advantage of their resilience to all these extreme conditions to regenerate, for example, sensors that use aptamers as the biorecognition molecule. You can raise the temperature, or you can use a solution that has a very low pH to get rid of the bound target analyte and regenerate the surface. Now, this is not that possible with antibodies, because if they are exposed to high temperatures, they unfold, they denature, for example, and then they cannot revert to their original native conformation. And lastly, modifications. How easy it is to introduce modifications. Granted, in both types of molecules, the introduction of modifications is relatively straightforward. However, in antibodies, in order to target specific regions, you have to go into a lot of, considerable amount of effort. You have to invest considerable amount of effort, and the results are not guaranteed, since any modification of the antibodies, even if it's site-oriented, is probably gonna cause some sort of damage to the 3D structure of the antibodies, therefore reducing their performance. Aptamers, on the other hand, can be modified site-specifically with high, it's very easy to introduce any type of modification and anywhere you want, and that makes them very attractive as molecules in biosensing, but also other applications where some sort of tagging is required, like the addition of a fluorophore or a functional group for their immobilization, and so on and so forth. So are they on a par to antibodies? That's the big question. I mean, that's how it all started, the big dilemma. I would say that we shouldn't be comparing aptamers to antibodies, because they are different. Their very nature is different. Antibodies are made up of amino acids. It is the properties that I'm going to discuss in the slides that are coming up, which make aptamers a lot better than antibodies in some aspects. And they shouldn't, in reality, be compared. They are meant for different purposes, and they offer different capabilities. Okay, so starting off with some advantages which are unique to aptamers. First of all, this base complementarity that we know exists between adenine and guanine and thymine and cytosine in DNA and uracil in RNA in place of thymine. Now, we can take advantage of that. We can extend a sequence, an aptameric sequence, and introduce overhangs which are complementary to other parts of to other sequences. So, for example, here I have included an example where aptamers have been combined into a polymer matrix made up of acrylate monomers, for example, to make this hydrogel, which is target responsive. So depending on the presence of an analyte, in this specific occasion it was cocaine, this hydrogel becomes less and less dense, if you want, because the aptamers recognize the target analyte, they break apart, and therefore these strands, these acrylate-based strands, get further and further apart, therefore making this hydrogel more and more porous. And now this target-induced change to the hydrogel has been exploited to actually develop a sensor where the liquid moves based on the porosity of the hydrogel itself. So that is for the signal transduction. How do you actually convert that change in porosity into something that can be easily detected, even visually, without the need of any specialized equipment? So I think that was a very interesting combination of aptamers with other molecules, and a nice signal transduction method that doesn't require any expensive piece of equipment. This same base complementarity allows them to acquire very complex structures. For example, DNA origami. DNA origami doesn't necessarily have to do with aptamers. It simply has to do with base complementarity of DNA. So you can see how a large amount of sequences can get self-organized into very complex 2D and 3D structures, which once combined with aptamers, I've got a nice example here. You can see this nanobox, as it's called, where the actual lock to unlock whatever there could be drug there for targeted drug delivery or for imaging, gets unlocked by the presence of an aptamer as the lock that recognizes a specific target analyte, in this case a protein. So the presence of the protein is what causes this shift in the sequence of the aptamer, unlocks the box that is entirely made up of nucleic acids. So that's very interesting, but it doesn't all have to be very organized and very, like this nano box that you saw here. Aptamers can also be used as a scaffold for the organization of smaller particles, nanoparticles, to form larger structures like these microparticles. So again, here the recognition of a target analyte is what causes the structural change to the aptamers. And since these aptamers are the scaffolds, then for these nanoparticles getting organized, but in no specific orientation like we had in DNA origami, then the whole structure of this microparticle changes over time, and always in relation to the presence and the concentration of the analyte being recognized. So you can have a very large, even visual change in the properties of this microparticle, which is made up of thousands of nanoparticles, simply because it is organized on a scaffold made of nucleic acids, functional nucleic acids, and aptamers. Okay, so obviously you understand that these sort of higher structures and types of sensing cannot be realized with antibodies, and that's why I'm saying that aptamers for me are a lot more than antibodies. But there is more. Their very nature, so their negatively charged phosphate backbone, or the bases, they make lots of specific and not so specific interactions, for example, that we have taken advantage of with 2D and 3D nanomaterials. The classic example being these gold nanoparticles. So what happens here is that aptamers kind of hug these gold nanoparticles in the absence of the analyte and protect them from the presence of salt, which cause aggregation. In the presence of a target analyte, the aptamers preferably bind to the analyte rather than hugging the nanoparticles, and that leaves them exposed to salt, which causes this aggregation and the shift in the absorption, which is visible even with the naked eye. You can see underneath the results from such an from such an assay where the gold nanoparticles got exposed to higher and higher concentrations of the analyte. The same holds true, in a very well-known and exploited phenomenon is the non-specific interaction of aptamers and nucleic acids in general with graphene oxide sheets, where they actually lie down on the graphene sheets or the graphene oxide sheets, and then they rise in the presence of their target. And this also has been extensively exploited for the fabrication of sensors. What is more, when it comes to their sequence, it is the very nature of the double helix or the presence of this negative phosphate backbone, which allows the realization of these smart ways of sensing or signal transduction, if you want. So, for example, there is a lot of dyes. Some of them were are very well known to biologists. So, for example, cybergreen or ethidium bromide that intercalate or bind to the grooves that are made by the double helix found in nucleic acids. So you can imagine an aptamer which is found, for example, like forming a stem, which is a double helix. Once recognizing its analyte, this double-stranded region gets disrupted. So if you had a dye intercalating or binding the grooves of this double helix, it can no longer bind to them, so you already have a sensor. The same holds true for the interaction, and this is a very relatively recent development of bases actually in this occasion, like thymine and cytosine. They are the most well exploited bases. Their interactions with noble metals like silver and gold. So again, these interactions can be exploited and can be taken advantage to create changes that are even visible with the naked eye and create very quickly a sensor for lots of different types of analytes. Last but not least, and again, based on the very nature of nucleic acids, this negative charge that aptamers have on their phosphate backbone has been ingeniously exploited in the fabrication of one of the most sensitive type of biosensor that exists, and I'm talking about field effect transistors. So there, in field effect transistors, you basically have to overcome this debye length to actually be able to sense. There is always this, anything that is beyond this length, which is just a few nanometers, cannot be seen, cannot be recognized by these very sensitive types of sensor. So you can imagine that if you have an antibody there, the fact that the antibody is about 10 nanometers high, it's already higher than the debye length, which means that the recognition of an analyte, a target analyte by the antibody, makes it really difficult for the sensor to actually detect this recognition. Whereas using aptamers not only gives you an advantage when it comes to size, because they are considerably smaller and they can be positioned within the debye length itself, and the same holds true for the recognition event. They also have the negative charge, which is very important for the actual functioning of the field effect transistors. So they're like the ideal molecules for this type of sensors, which I'm sure we're gonna hear more and more about them in the near future, exactly because of their extreme sensitivity. The same holds true for real-time detection of analytes. Something which is very difficult with antibodies. So aptamers, again, they can change their conformation in the presence of the analyte, And here you see a redox probe attached at the end of the aptamer, which gets closer to the surface of an electrode, in this occasion, in real time, transmitting information to changing the electrochemical properties in real time, something, again, which is difficult with antibodies, especially when it comes to small molecules. The whole, basis of nucleic acids has been exploited by biologists to set up PCR assays, techniques that are very common in usual molecular biology laboratories. It is only recently that we have started taking advantage of techniques like that, the amplification, for example, of nucleic acids, and incorporating them, integrating them into sensing approaches and sensing principles. So, for example, here you see an aptamer coated slide or a surface where aptamer has been immobilized, and then these aptamers, if the target analyte is present, they bind to the target analyte. If there isn't, then they bind to this circular DNA, and we have an amplification through rolling cycle. It's called rolling cycle amplification because it only makes use of one enzyme. It doesn't require the sequential steps of double-stranded DNA denaturation, primer annealing, primer extension, which are common to PCR. It's something that renders itself nicely to portable assays. So this is yet another approach to amplify the signal, and you can imagine that these little dark dots that you see are molecules for example, gold nanoparticles or biotin molecules that will allow the amplification of the signal simply due to the nature of the recognition molecule itself, which is, at the end of the day, a nucleic acid itself. So what's the catch? I mean, I have been talking so far about all these advantages of aptamers and how nicely they compare to antibodies. And yes, it is true that the number of publications has been increasing, on a steady, or I should say on an exponential basis as years go by. I mean, this is an image I got from a recent review on aptamers, and it is categorized based on the search term, basically, aptamer plus drug delivery plus biosensor, or aptamer in general. So you can see the trend. And the image I've got on the right-hand side just shows the expected market, the anticipated market, or the market forecast for aptamers globally up to the year 2028. So it all looks very promising. So you would be I guess you're wondering, why haven't you heard about aptamers and aptamer-based sensors, or aptamers being used in more and more applications? The reason is that, yes, they are promising, and yes, we have taken advantage of a lot of their unique features offered by the nature of aptamers. However, there are some limitations. For example, I mean, one of the main ones being the fact that we only have to work with 4 letters. So you can imagine a language where you have to set to create new words using just 4 letters. There aren't that many combinations that can arise from this 4-letter language. And I'm talking about adenine, guanine, thymine, and cytosine in the case of DNA aptamers. Whereas antibodies make use of 20 different amino acids, some of them being hydrophobic, something that doesn't exist in nucleic acids. So there are efforts, there's a lot of effort being invested recently in introducing modified nucleotides so that we mimic, let's say, the word of antibodies and amino acids, the flexibility and the versatility offered by these 20 different amino acids that we have there. So the modifications can be introduced in the bases themselves, and here you see some examples, or the backbone of the molecule itself. So there is lots and lots of modification. Problem is that the moment you start introducing these modifications, you lose on the ability to amplify the sequences using traditional techniques, using traditional polymerases, simply because the polymerases cannot work with a lot of these modified and nucleotides or backbones. So you gain something, you lose something. The other thing is that all these modifications do come at a cost. So there is a certain need to actually expand on the letters that we use in this language of aptamers and functional nucleic acids, but it does come at a cost. I'm sure that in the future, these modifications are going to be cheaper, and there are already polymerases that can amplify sequences that are modified, and I expect their prices to also go down as well in the future. Then some issues with Selex itself. Depending, and there is a lot of effort as well, as I showed you the different variants, but also selecting the library. That's another thing that has received a lot of attention, the library of random oligonucleotides to start with. So the selection can be biased. Instead of going for a random oligonucleotide sequence, a random library of oligonucleotide sequences, these libraries the libraries that we now use are biased. We introduce on purpose bases at a higher percentage simply because we know that there's gonna be the tendency to have a 2D or a 3D conformation, which is absolutely necessary for the recognition of any target analyte. The same holds true with how you manipulate the primer region, the primer binding sites, and so on. I don't want to tire you too much about this, but in any case, starting off with an appropriately designed library and having a very good workflow when it comes to the selection of the sequence, the initial selection of the sequence, is of paramount importance. Finally, validating the selected sequences. That, to me, is the biggest problem that aptamers currently face. And I've put here two recent publications which report exactly that. The aptamers do not bind their target analyte. I'm talking about specific aptamers which have been reported by many scientists in the literature as being able to bind selectively to a target sequence. Now, the reason for this non-affinity or non-ability to bind to a target sequence, and you might be wondering, so how on earth did they manage to publish, or what sort of results they got? The thing is that it all has to do with how you assess and how you validate the sequences that you have selected for. So it does take, traditionally, aptamers have been exploited and they have been developed by people working on sensors. So usually the techniques that have been used to characterize a sequence on whether it binds to an analyte or not are prone to mistakes, are prone to non-specific binding, are prone or are very vulnerable, very susceptible to give you false results. So it is of paramount importance to actually validate existing sequences as well as the ones that are newly selected with techniques that are more standard, like the isothermal titration calorimetry or SPR, or, you know, it's kind of complicated, but even with NMR. So there are ways to validate the sequence, and one has to go into that effort because there is the need to know whether there is specific binding for the target analyte. That's, for example, that's on one hand, and on the other hand, one has to optimize the buffer that is gonna be used in setting up an assay. Here I have an example that from the results that we got from a very well-studied aptamer that against ochratoxin A. So people You can see this is reported in the literature. What you have in front of you are spectra from circular dichroism. So in the literature, what is reported is that the aptamer cannot bind to its target analyte, ochratoxin A, in the absence of divalent cations like magnesium or calcium. Fair enough. And we have also seen the same thing. You can see the spectrum. Here you have polarized light passing through an aptamer solution, and based on the chirality of the molecule, you see differences in the spectra. So you can see no difference in the spectra here in the absence, in the presence of a buffer that doesn't have any divalent cation. So what people actually use, the majority of the publications actually report this buffer, which has magnesium added to it. And as you can see, there is a change in the spectrum relative to the buffer where magnesium is not present. That means that the aptamer has acquired a different conformation, and you can see that it actually binds to its target analyte. However, the interesting thing that we found is that this conformation that the aptamer has, which is different in the absence of the divalent cation, is there even in the absence of its target analyte. So that's why you don't get to see any differences in the spectra here. So if you were to design a sensor or an assay, and you wanted a large structural conformation from your aptamer only in the presence of the analyte, which you are to exploit, let's say, to enhance your signal, or to have some sort of magnified signal transduction, you would like your aptamer to only be able to acquire this new conformation in the presence of the analyte. So what we saw was that if you don't add magnesium, but you put calcium, then your aptamer in the absence of the analyte doesn't have this very important G quadruplex structure, and it only acquires that in the presence of the analyte. Now, this is very important because that allows us to create, to develop assays which have this all-important structural conformation. And for example, I have an example here on an aptamer that we have also been working, the aptamer against aflatoxin M1. So for these aptamers, another signal transduction methodology which magnifies the signal is this triple helix molecular switch. So you have a sequence in the middle, the aptamer sequence is the green one, and you have two extensions of the aptamer hybridizing under special conditions with this sequence in the middle. So if your aptamer, what we've realized is that if your aptamer, if we take the original aptamer, that's the original aptamer for aflatoxin M1, and we add these extensions, the whole approach doesn't work simply because there isn't a large enough structural conformational change. However, if we truncate the aptamer, like we've done here, and we add these extensions, then we've managed to convert an aptamer that is not meant to undergo a large structural conformation into one that the binding of the target induces a large enough, I wouldn't say structural conformation, but let's say strain, puts enough strain in the sequence to actually make this triple helix molecular switch work. So there is bottom line, and as a summary, depending on the aptamer that you have and the specific considerations of your assay, you really need to understand whether there is the affinity there to start with, and then optimize the sequence so that it works for your, sorry, there's a bit of background noise. Okay, so you need to optimize this. You need to optimize the sequence to make your assay work reproducibly and provide you with a large enough signal to quantify. finally, this is the latest development. As you can understand, all this is based on a trial and error. This rational design, no matter how good you are or what sort of equipment you've got to actually decipher the 3D structure of an aptamer and understand how the buffer composition and ionic strength affect this structure, there is simply too many sequences and there's simply too many experiments to be undertaken. So ideally, one thing that would be good would be to actually speed up this process by using simulations. So we have also developed within the context of next-gen microfluidics a process flow to simulate all this and minimize the search space of these sequences and the possible modifications and mutations that you can introduce. It is early days because simply the computing power is there, but the algorithms, the force fields required to run this are not refined enough as they are for proteins. But we are working on it along with other colleagues from other research institutes. So we expect and anticipate some really exciting developments in the near future when it comes to molecular dynamics and docking experiments and simulations of aptamers with their target analytes. So applications, I've already shown you quite a lot. I just have this one slide. So obviously aptamers are involved in biosensing of small molecules, but also cells. So for example, they have been used in microfluidic platforms to isolate or enrich circulating tumor cells. But they also have lots and lots of other applications, for example, as immunomodulators in immunotherapies and cancer therapies, chemotherapies, as agents for imaging or delivery, targeted delivery of drugs. So the possibilities are endless. One thing that I think has become apparent is that you really have to get your aptamer sequence right, depending on the application that you're interested in, and design the assay in such a way so that it works for the signal transduction and for the purpose that you want the assay to work for. So if it is, for example, for the assay, if the aptamer is meant to bind to cells, then it is worth investing in performing whole cell selects, because if you isolate or if you use an aptamer that has been selected against a recombinant antigen, which you expect to find on the cell surface, then it's pretty certain that it's not going to work the same when you use the same aptamer that has been developed with a recombinant antigen to bind to a whole cell. Yeah, I mean, that's the take-home message. Get your aptamer right and design your assays smartly, depending on the application. I mean, I could go on and on and on when it comes to different applications, but I think having seen already all these interesting developments on how aptamers can be combined with nanomaterials, how they can be integrated in microfluidic devices, it has become apparent that the possibilities are there, but the challenges are there too, and one has to invest a bit in the beginning in order to have the whole process right to actually get it the application going. So a couple of slides in the end on our lab, which is located. This is what you have in front of you is the Bioacademy, which is 4 or 5 kilometers away from the city center of Athens, Greece, next to a mountain. We've got a nice forest next to us. And our lab is found on the first floor in that building, exactly where the circle is. So when it comes to our activities, I've talked extensively about aptamers and developing assays using aptamers and the biophysical characterization of aptamers that we are undertaking so that we develop, customize, and optimize assays for biosensors and other applications based on aptamers, but we're also involved in the development of appropriate chemistries, which truth is they're equally important when it comes to developing an assay, especially with aptamers. Aptamers are notorious for being extremely sensitive to the way, for example, that you immobilize them onto the surface. They're very sensitive to the type of linker, the length of the linker, and the nature of the linker that you're going to use to immobilize them on a surface, be it on an electrode or being on a thermoplast foil like we have in next-gen microfluidics. So it really pays off to actually combine the biochemistry with the surface and interfacial chemistry to get an assay that is truly robust and reproducible. Other than that, our main focus as a lab when it comes to surface chemistry is the development of material-independent chemistries to not only biofunctionalize microfluidic cartridges, sensors in general, but also to tailor their surface properties and render them anti-fouling or tailor their wettability and so on and so forth. So that was my last slide. I hope I didn't tire you that much. So thank you very much for joining once again. And if you've got any questions, Divesh has a couple of slides more. I don't know, Divesh, should we
No,
have
let's go through the questions.
Okay, sure.
First, yeah, it's just slide is just to close and to
Yeah.
remind who we are and what are we here for. But George, thank you very much. That was a great overview of aptamers, their uses, their benefits, methods. That was very useful for me. And from the amount of clapping that I see that it's also been useful for many others.
I don't get to see the clapping. I just see this thank you. So perhaps I should.
Well, it's a lot of clapping. It's a lot of clapping. Yeah. Okay, so I think we have a few minutes for questions. So feel free to, I mean, unless everyone is going to ask questions at the same time, feel free to unmute yourself or raise your hand to ask a question or simply write it in the chat. Do we have any questions from anyone? Otherwise, I will ask some questions.
It's probably too long, so.
Oh, look, we have a hand up.
Oh.
Nastasia, feel free to unmute yourself and
Hi, George.
Hello.
Thank you for a great lecture. I was wondering, you said it's important to get the aptamers right. How long does such a selection process usually take?
It really depends on the target analyte and the technique that you're going to use. So if you go for the traditional selects where you immobilize the analyte, and given that the analyte is, let's say, hydrophilic, then it should take something between 2 to 3 months. If you use capillary techniques like the capillary electrophoresis or the magnetic bead, you can greatly reduce the time required to a month, maximum 2 months. That is all considering that everything goes according to plan, or that you don't have to work with a target which is difficult. So, for example, if one searches on what sort of aptamers and for what sort of analytes are out there, you'll notice that most of the analytes are hydrophilic. And that is because targeting hydrophobic analytes with these functional nucleic acids and aptamers, which, their very nature is hydrophilic, is difficult. There are ways to circumvent that even without the use of modified bases, but it makes the selection process lengthier. But yeah, it all has to do with the technique, the target, and the equipment. I'd say it should range from, a minimum of 1 month, and it can take, to get something for targeting cells, it could take up to a year.
Thank you very much.
You're welcome.
As a follow-up question to that, perhaps, are you familiar or do you know of microfluidic methods to perform selects. Do you think
Yeah.
that that is a good, perhaps a way to accelerate it or to make it more efficient?
Absolutely. For example, this microfluidic, this capillary selects is actually being undertaken in microfluidic cartridges. So the latest development is that there is I've read reports where they've even used digital microfluidics to actually perform this, because obviously minimizing the space and being able and coupling it with magnetic beads, right? So it's a DMF coupled magnetic beads selects, obviously speeds up the process. Problem with that is that not a lot of researchers actually have access to all this. But yes, there are ways, and obviously microfluidics offers lots and lots of advantages when it comes to the selection, not to mention the application of the aptamers themselves, yes.
Okay, thanks. Do we have any other questions from anyone else? Yes, please, Marcus.
Yeah, hello, George,
Hello.
for your nice talk. It was very good overview. Do you make selection by yourself, or do you use only aptamers you find in the literature?
Yes. So far, I mean, we have been working extensively with existing sequences, which in itself is enough of a challenge as it is to get the right technique and combination, I would say, of techniques to actually characterize and post-selects optimize them. We are recently endeavoring into the selection of aptamers. Our focus is always on the small molecule analytes.
Then you want to use capture selects for that?
Yeah, capture selects or capillary. Yeah, capillary electrophoresis, yes.
So I have only an offer for you. We developed aptamers in our lab. So if you're interested in that, we can be in contact.
Yeah.
Or if you are interested in other aptamers for other targets for that.
Sure. I can't see you from where my colleague Zena came in.
No, I get an external man get me a link. So I'm Markus Menger from Fraunhofer Institute in Potsdam, Germany.
That you organized the nice workshop in the summer, right?
That's right. Yes.
Yeah. Okay. Yeah, sure. I mean, I'd be more than happy. Yeah.
Yeah, I have one comment to your slide 2020, I think, where you discuss how is the reason that aptamers not bind yet that you find in the literature.
Not 20. You mean this one?
Yes. No. Yeah. This one. Yes. Yeah.
Yeah. 22.
So what we found in our labs that mostly the problem is that aptamers you find in literature that not bind to a target is that the conditions are very not the same that you want to use in your application, and you must mention that aptamers make a specific 3D structure, and that is depends on the condition of the buffer. And when the selection process was make a different buffer, then you get changes in the conformation, and then that can be a reason why it doesn't work in your assay. That is one main reason, and the other reason is depends on the target. Perhaps when people use a protein as a target and they make the synthesis of that protein by example E. coli expression, then they want to use the aptamer against the nature target molecule, and that has post-translational modifications, then you can get problems in binding, of course, because you don't use the right surface for the aptamer selection. And that I must a little bit say different to you. I think aptamers are very specific and sometimes more specific than antibodies, because of that, because they can really recognize a specific surface of a target molecule.
Yeah, it really depends. For example, why I said if you have an aptamer, for example, that binds to a circulating biomarker to a protein, yeah, then usually the selection will just involve using a recombinant form of the protein or something like that. But when you actually get to use it, apart from worrying about degradation by exonucleases, it could also bind non-specifically to a lot of things. That's why, of course, they are selective and specific, but it all really depends on the matrix that they are exposed to. And I was really happy to hear when my colleague came over to the workshop that you organized in the summer that you also have this belief that we should do more about proving that aptamers work. And I agree that the buffer is one thing for sure. But for example, you know, I don't want to go and mention specific aptamers that we have tested, but it's not just the buffer, because we have used lots of different buffers, the selection buffer as well as lots of other buffers, and some sequences that are reported over and over again in the literature, they simply don't work. And the reason for this is that they have only been, their binding has only been validated by assays like the gold nanoparticle, for example, that I showed you, which is notorious for, you know, non-specific interactions of the gold nanoparticles with the target itself. You can have protection of the nanoparticles, you know, just by the target. So it really, as you also said during the workshop, it is really worth investing in using more than one validation methodology to actually prove the performance and establish the performance of your aptamer prior to using it for a downstream application. And I think we are on the same page that there.
Yeah, you are right. So I also support your opinion that you should, before you use an aptamer, you should try it in different methodologies to see that it's bind in different methodologies. And the other thing to prevent this difficult or this hindrance, the best is you make an adaptation of your selection buffer to your application buffer.
Absolutely.
Then you can prevent this problems a little bit.
Absolutely. Which only very few groups, I haven't seen many such publications where the selection has actually been undertaken in the buffer or the solution where they actually want the aptamer to be used. Yeah, you're right.
Yeah, it's only a comment, but thanks.
You're absolutely right. Thank you for joining.
Yeah, thanks for your talk.
Sorry. Oh, I just had a comment with lots of ramifications, which is exactly what we want. Christoph, do you have a question, comment? Please feel free to just go ahead.
Do you hear me?
Yeah.
Yeah. Hi. Hi, George. Thank you very much for this very interesting lecture. I think 1 year ago I wouldn't have understood that much, but lately I had some friends who told me a lot of stuff about aptamers, so I could follow quite good. My question, at the end of your talk, you were showing us some results about the simulation. And what is the stage of your results? Do you can apply it already, and do you have like as I understand you can use it to be faster in the SILEX process, right?
We cannot, truth be said, we tried using it for the de novo selection of sequences against analytes. You know, it doesn't work because the force fields are not there yet for that. So we can only use it as it is, and we've done it. We've validated that against known aptamer sequences that have been, and their interactions with their target analyte, for example, the thrombin aptamer, the ATP aptamer, and so on. whose structures and their interactions with the target analyte have been resolved either by crystallography or NMR. So we did get the same sort of results using molecular dynamics, following lots of iteration rounds when it comes to refinement of the algorithms and so on. It is simply not there. I mean, ideally, we would also like to proceed to the selection of new novel aptamer sequences without having to undergo select. So that's one thing. Another thing is that we would like to use this to actually speed up the process of post-select optimization. Therefore, to have a genetic algorithm, right, introduce mutations and things like that and score them. It does to an extent, but with a lot of uncertainty. The thing is that it gets better and better because the software where all this is based, or actually the combination of software programs, where each of these steps required for this process are based upon, are improved as we speak. So that's why I said that I anticipate that at some point, this whole molecular dynamics and simulation-based design and optimization of aptamers will improve tremendously to the point that the antibody and drug discovery, for example, all these interactions are when it comes to proteins. But we're not there yet when it comes to de novo selection, unfortunately.
Okay, yeah.
It would be very good, but no.
Yeah, so there is some more effort to put on this, and you will improve by further projects on this, right?
Yeah, I mean, it requires also a lot of computing power.
Yeah,
as you can understand,
guess,
and we have access to the hypercomputer, let's say, of the Greek state.
Oh, wow.
So we managed to get access to that, but even there they need a standard protocol which runs uninterrupted, right? So every now and then there is a glitch coming up, which you have to intervene, right? And they don't give you access to this high computing power if you don't have an already set program that you're gonna run or process, right?
Yeah,
which is
I know.
defies the purpose, right? But anyway.
Wow.
Yeah.
Okay. Yeah. Thank you.
Thank you.
Thank you very much. Marcus, do we have an extra question?
Yeah, can you integrate modifications of the aptamers in your software? So for the different nucleotides, is that possible, or is just an ongoing process?
No, unfortunately not at the moment. No.
Yeah. I see.
That would be very interesting. Yeah.
Yeah. Alternative or an addition to that modeling is, of course, to use dope selects after you have find an aptamer for getting better affinity or specificity to a target molecule. But that is more a practical way then.
Yeah. As you said, this is all nice and good. There are a lot of efforts, and I see a lot of publications coming up recently where they use some sort of, or one sort or the other of simulation up to a different level, for example, up to the docking, or they proceed with the molecular dynamics. But the truth is that the computing power is there, the knowledge is there. It's the algorithms and the programs that kind of fail us for the moment. I think they need a bit more training when it comes to, providing them with input from already resolved sequences and how they interact with target analytes. For that, we need NMR or crystallographic data, and we simply don't have enough. That's the problem. It's still early days. I mean, you've seen how many publications there are out there with thrombin, cocaine, ochratoxin A, and ATP. It's always the same aptamers, right?
You're absolutely right. Always the same aptamers and
always
targets.
Always the same aptamers. It kind of gets tiring.
Yeah, mostly.
Yeah.
Yeah, that's the problem. You are absolutely right. You need more data to use KE for get better results in that case. Yeah.
Yeah.
Interesting.
Yeah, we need more bioinformatic people in this field.
And more people working on aptamers in general.
That's right. Yeah.
Okay. It's just funny to hear sometimes, because every field of biochemical study seems to have this one model. You know, when you work with membrane biophysics, everything is alpha hemolysin, alpha hemolysin, alpha hemolysin. It's like, have some imagination. There's lots of membrane proteins.
Yeah. There are a lot of unknowns though. I mean, the fact that there's so many publications still coming out on the same molecules just goes to prove how much we don't know about aptamers and how exciting the field is.
Well, your excitement is certainly contagious, George. Okay, I would assume that we have concluded with questions, but of course, I think we have time for a last one. If there's anyone, okay, I'm going to take that as a no. So I'm going to take the chance to thank George again for a very interesting
Thank you.
presentation, and also for those who have asked questions and raised interesting points of discussion, and of course for everyone else for joining the webinar. Yep, so prior to ending, I would just like to remind, in the context of Microfluidics Innovation Hub, if you're interested in the kind of services or technologies that George has described throughout his presentation, or other technologies and expertise that he has in surface modification in terms of developing a microfluidic application, I would like to remind you that we still have our open call funding available, which is to develop a microfluidic product. Let's say, for example, for in vitro diagnostics from TRL4 to above, where at the MIH we will combine the technologies and expertise of companies, perhaps like George, who work on the assay transfer, but others that develop microfluidic circuits, that manufacture them, that do spotting of reagents, that develop readout devices. So if there's anyone here that is interested in this kind of funding for any kind of application, you know where to find us. Our website is there with additional information on the open call. Other than that, I would thank everyone again for joining, and we will let you know when we have our next webinar. Other than that, again, thank you very much, and enjoy your evening, and see you next time.
Thank you.
Thank you, everyone.
Bye.